Hi, I'm Michael Hoffman, and today I'll be talking about how human papillomavirus, or HPV, changes the host epigenome and transcriptome to promote tumorigenesis. I have no disclosures for this talk. So the standard model of HPV gene regulation involves the virus getting episomes, small, about 8,000 base pair chromosomes, into the cytosol of individual cells. And there's a gene expression program where these so-called early genes are first expressed, especially E6 and E7, which are transcription factors that are responsible for driving the rest of the HPV gene expression program. So my PhD student, Mehran Karimzadeh, who did all of the work that I'll show you from now on, was looking at a number of cervical carcinoma and head and neck carcinoma samples with HPV and looking for these HPV transcripts, and found that in E6 and E7, there's often high transcription, but in a small number of cases, there's actually a relatively large number of cases their E6 and E7 expression is low, and he wanted to investigate why that was. So if this led him to wonder whether there were other factors other than E6 or E7 promoting oncogenesis. If E6 and E7 aren't expressed in some patient tumors, what other factors can contribute? So he considered that HPV integration is common to most HPV tumors, whereas non-integrated episomal HPV is common to most non-malignant warts. So his hypothesis was that other factors promoting HPV oncogenesis might have something to do with HPV integration. We should note it's still possible that E6 and E7 were active and initiated tumorigenesis, but became inactive in later stages. So working with Christopher Arledge and Ariana Rostami and the Monty Lupian and Scott Bratman labs, we conducted a multi-omic study of 5 HPV positive cell lines. These were all derived from cervical cancer patients, and we identified HPV integration sites using our software, which used to be called Bellerophon, but is now called Polyitis, and investigated the transcriptome and epigenome at the site of HPV integration, doing RNA-seq, ATAC-seq, and ChIP-seq for CTCF in these cell lines. So let me show you an example of the sort of thing that we found. Here's one cell line, HMS 001, where there is an HPV integration site at approximately 47 million base pairs on chromosome 20. What we did is we looked at this integration site, which occurs only in this cell line, and we looked to see whether there were changes in the epigenome around it. So you might see qualitatively that comparing this to the other 4 cell lines that don't have an integration site at this location, you see a change in the sort of peaks, ATAC-seq peaks that are extant. In fact, you can see at the bottom that there's often quite a big difference between the extreme value of the other 4 cell types and of what we find in HMS 001. We display this on a slightly different scale so that you can see a heat map of each one of these peaks, and it maps uniformly to the genomic position. And we can do the same sort of thing for our CTCF ChIP-seq results, where you can see, for example, here's a ChIP-seq peak that you find near the integration site only in the cell line that has the integration site. And you can find other things like peaks that don't exist there but do exist in the other cell types. That's fine qualitatively, but we also wanted to do the same sort of thing and control for any sort of random effects that we might see. And so one thing that we did is we took all of the integration sites we found in these 5 different cell types, right, and we scrambled them, and then we analyzed the data around the original locations of the integration sites and also scrambled locations on the same chromosome where we didn't actually have an integration site to see whether, is the integration site that was actually affecting things. What we found looking at various assay types is that very close, so within 1,000 base pairs of a integration site, we have a big increase in RNA-seq shown transcription compared to especially permuted negative controls, right? And we see some slight increase in open chromatin, and this sort of effect is seen in an even bigger way at the 1,000 to 10,000 base pair range, where you see an effect for all of these different assays. And if you look at bigger than 10,000 base pairs, the effect kind of goes away as you get farther and farther from the integration site. So we wondered why this effect might actually be seen. One thing that we considered is that CTCF binds to HPV-16. So unlike a lot of other viruses that integrate into the human genome, a unique feature of HPV is that there is a CTCF binding site. And in fact, this is well conserved across a number of different HPV subtypes, right? So HPV-16, which is most of what we're looking at here, has it, but you also have a good motif match in lots of other cell types, although we do not have it in a small number of HPV cell types, which will be useful later on as I can show you. With our ATAC-seq and CTCF data, ChIP-seq data, as well as the RNA-seq data, we can characterize the epigenome and transcriptome not just of the human genome, but also of the HPV genome as well. And so here you can see, for example, that in all 5 of these cell types, we do have what looks to be like a CTCF ChIP-seq peak, and we do have varying amounts of open chromatin shown in the various cell types. One other thing you need to consider here is that the HPV genome is circular, right? And the integration doesn't always occur at what is position 0 on this reference genome. You can have integrations at all sorts of places. And that's what these various dotted and dashed lines indicate, where in this particular cell line, like CASCI, what part of the HPV-16 genome do you actually find in the middle of the integration site? So the main summary so far is that the main cause of HPV tumorigenesis is thought to be E6, expression, but while these genes have a low expression in many patients, HPV's strong CTCF motif and our ChIP-seq results indicate strong binding of CTCF. So this suggests that this may alter the native genome organization of the host. Are these able to promote tumorigenesis? So one thing that we looked at is the expression of genes around HPV integration sites. So you can see in all 5 of our different cell lines, you can see all the different integration sites here, right? And you can see that some, like HMS001, only have 1 integration site, while 93VU147T has a whole bunch of them. We looked at all of the genes that were within 500 kb and looked at the RNA-seq transcripts per million for that cell type against the average for all of the other four cell types to see whether there were big changes in the cell type where there's a nearby integration site, right? So these so-called outlier changes where we have at least 2-fold difference of expression, right? And the TPM difference is at least twice the standard deviation amongst the 5. And those are where shown in colored non-transparent blocks here. So I can zoom in on 5 of these so you can get a better example of the sort of things that we see. One for each of our 5 cell types. You can see, for example, the SEC090, the FOXY1 gene, which is 60 kb away from the integration site, or one integration site, has very high expression, and the other 4 cell types have very low expression, right? And this is something that, a pattern that you see over and over again. Here's one CHA where you have low expression versus high expression in most of the other ones. And you can see the same sort of thing here and here. And of course, these are very nice cherry-picked examples, but does this trend hold in a more rigorous fashion? I would like to think yes, and the way that we have shown this is by doing a permutation analysis, somewhat like what I discussed before. It's not. This is something that shows how robust the outlier analysis is to where we set the threshold for what an outlier gene is. And basically, you see that within 100 kb, there are always outlier genes at each threshold in the real data, which is red, versus the permuted data for a wide range of deciding what outlier is. And when you look at larger regions within 500 kb, this mostly collapses. So basically what we keep seeing is that the HPV integration gives every sign of having local effects on transcriptome, but we don't see it at much further away. So we decided to take these results in our cell lines and look in cancer tissues from TCGA as well, right? And we find very similar patterns in head and neck squamous carcinoma and cervical carcinoma, that we find outlier genes near integration sites that we have detected in all of these TCGA samples with our polyitis software. Another thing we wanted to look at is it something related to HPV specifically, or is any viral integration capable of causing outlier gene expression? So for this, we decided to look at Epstein-Barr virus and do a similar analysis. Luckily, there's a lot of data around with Epstein-Barr virus integration, a lot of it from ENCODE and elsewhere. And basically what we see is pretty much a null effect all the way across the board. You can see, insignificant p-values, certainly going to be insignificant if you correct for multiple testing and so on. And if you compare, on a by distance to integration site basis, you can see that, within 100 kb and especially within 50 kb, you see quite significant changes in transcription, as well as open chromatin and CTCF binding when you're close to an HPV integration site. But when we do the same sort of thing with Epstein-Barr virus, you don't see it. Finally, one thing we wanted to look at is, does CTCF binding to HPV, is that what causes the outlier gene expression? And for that, the cell lines, the HPV variants that don't have CTCF that I mentioned earlier in this talk come in useful. So the HPV 73 strain lacks the CTCF sequence motif, and while there are small number of HPV 73 integrations in TCGA data, none of these integrations show any sort of outlier gene expression, which is consistent with our model of the CTCF binding site in HPV being responsible for some epigenome remodeling. So basically our model is based on the fact that CTCF can affect topological structure of chromatin, and that can dramatically change expression of local genes. So that's what we think is happening, that the CTCF binding site, when integrated, can affect gene regulation nearby and can also affect alternative splicing, which I didn't have time to talk about today, but it is in our preprint, which you can find on bioRxiv. It's Karimzadeh et al. And is available at that address. And to quickly summarize, we discovered that CTCF binds to a conserved sequence motif in HPV, and HPV integration modifies the local epigenome, possibly reorganizing topologically associated domains. I'd like to thank my lab. I especially like to thank Mayron, who did the work presented here. Thank our collaborators at Princess Margaret Cancer Center. And finally, I'd like to thank all of you for your kind attention. Thank you very much.